As I said, I'm going to have such sessions now, always at the beginning, the first sort of 10 minutes or something. Okay, so today we talk about, in fact, secondary structure prediction. Oops. The next Tuesday is Whitsun holiday. Remember that? It's a holiday from the university. So the recap: in principle, sequence determines structure, determines function. In principle, you throw the protein into a solvent, it adopts the same unique three-dimensional structure. And in principle, we should be able, therefore, to from sequence predict structure and function. We cannot do that because the problem is too complex. We have these alignment methods, global, local, that work at some region where it's simple, high similarity between two proteins, sequence alignments, sequence profile alignments, and profile alignments go further. Protein structure prediction, we can only really do homology modeling, and the CASP experiment is one way to test how good structure prediction works. Secondary structure is a sort of macromolecular substructure of a structure that you can easily see, that happens in all proteins, essentially. The major two segments are alpha helices and beta strands. And they are defined historically by hydrogen bond formation. Linus Pauling brought that up. And the reach of comparative modeling is that we roughly can model, comparative modeling can sort of reach half of the pie somehow. This is the percentage of proteins, and the blue here, this is the dark blue, the experimental. The blue depends on how good your model is supposed to be. So that is why there's sort of a grade here. On the level of per residue, the number is a little bit lower. What that means is that for many proteins we can in fact map a fragment, but we cannot map everything. The good news is comparative modeling predicts for many 25 million proteins at the moment somehow structure, and that part of the good news is at 100K per structure, you rightfully said it's expensive. This corresponds to something like 3 trillion. That's more than the GDP of England and France at this point. That we gain by doing computational biology. Essentially, if we would have to do these structures without a computer, then that's what you would pay. It's amazing what computers can do. The bad news is, for most proteins, comparative modeling cannot be applied. For most proteins, in fact, we don't have a structure that is similar, cannot recognize it, or there's a region where we cannot do that. And that means that we have to get rid, or we have to approach the problem on a different level. 3D is no longer doable. I will talk about 2D and I will talk about 1D, and I begin with 1D because that's the simplest. And the first example of 1D is secondary structure prediction. About words, secondary structure, some people sort of believe or use the word to assume that it's 2D. Again, it's a string. It's intrinsically one-dimensional information that you have to remember. And the way it's defined is by hydrogen bonds. For instance, in DSSP, there are different types in DSSP. Typically, what concerns you is there are helices, strands, and there's something else. There's something else. Sometimes it's called loop, and sometimes has other names. Essentially, this something else is just other. And the reason is helix is well defined, strand is well defined, other is not. It's the bunch of all the things I cannot describe. Okay? Now, local sequence determines secondary structure. Here is an old paper from 30 years ago, in which Kapsch found out that some stretches are always observed in a helix, others always in a strand, and others in non-regular structure, or in other. Here again call it loop. And again, this is one of those things. We call it loop simply because that's easier to say. It's misleading. And you see here some peptides, pentapeptides, so 5 residues long, that are observed in strand and helix. The goal of secondary structure prediction methods then is to simply take a sequence and sort it into these 3 bins. One question is, how come that pentapeptides can be observed in strand and helices. How can that happen at all? I said that, you take this particular sequence, you throw it into a solvent, and it adopts a unique three-dimensional structure. So if you have the same pentapeptide in completely two structural conformations, beta strand and alpha helix, then my statement appears to be invalidated.
Probably it depends on the context.
Yes, that's exactly the point. So bless you, the protein folds with many more than 5 residues, and the context may, in fact, you could imagine that in fact, it's not only that you can imagine, there are examples for that, there are many examples for that even, where if you have an isolated string, it starts making a helix, because what is next to you is the hydrogen bond I+4, and the energetic, right, correct, or easy solution is the helix. Then you see that actually next to you in 3D comes another strand. And that, in fact, is a stronger preference in this particular case, maybe. For that particular sequence is such that forming a sheet with another strand would energetically be even better. So it flops or flips from a helix, an isolated helix, into a sheet, just according to its context. And indeed, so the sequence always forms a unique structure, but the sequence is longer than 5 residues. 10 residues is still very short. Now, a simple method then to predict secondary structure would be to take the known structures, find the longest consecutive run of stretch of sequence, and simply ask where it occurs. So the longest run that occurs only in helices, the longest run that occurs only in strands, the longest run that occurs only in the other state, build up a data set, and you have a structure prediction method. Let's call it a database. You have all the fragments that are unique to one of these three. What's the problem with that? Well, let's begin differently. Would that work at all? Let's turn that around. So what you have to learn as a scientist is you come up with an observation, that is your observation, and you have to now find reasons why that is wrong or right. So I'm going to say, this is not going to work. Explain to me why. Or if you want, you can also answer the question, or the statement, it's going to work. Pick whatever you want of these two and kill it. Again, this is what we have to learn as scientists. Yeah? Sorry.
We have helix strands and loops, so the third structure is not well defined, so we cannot really compare that.
So the statement is that we have helix, strand, and other, or loop, and maybe for one particular stretch the structure is not well defined. Is that what you said?
Yeah, that's third part of it.
Oh, okay. So I see what you say. So now I believe what you say is, I'm sorry, I do not see what you say. I assume what you say is that there are helices that are well defined, there are strands that are well defined, but loop is not well defined, so it would work for helix and strand. Is that what you say? Okay, if it would work for helix and strand, if I gave you a method that predicted helix and strand right, what about the other state? Can anybody answer that? So I said that other is not well defined in terms of structure. So that is true, and that's essentially all you say, and it's absolutely true. But if I had a prediction method that gets alpha right, beta right, how well would that method do on other?
It will give others very predictably as well.
It will give others very predictively what?
If A and B and C are three components, if I can identify A and B, then I can identify C as well.
Yes, it's a zero-sum game. So if, in fact, I have 3 states, and if 2 of those states I can recognize correctly, then I get the third one by exclusion. It's the only one that is left. It's true. So if I could do alpha and beta, then I could do loop. Back to my question, can I do alpha and beta, or can I do it? So what would work and what wouldn't work? Yeah?
The answer is it will depend on the context, so it will not always be the
That is true. So that brings us back to the issue of the pentapeptides. My algorithm now is a little bit cleverer. This is 30 years ago. But my algorithm now says, well, if I encounter pentapeptides like that, I'm not going to use them. I have a larger database now than they had in those days. In fact, they had much less than 100 proteins. And for my larger database, I can just look at whatever chimer I find to the highest and shortest length that is unique in the database. That is not, so these are the throwaway candidates. All I'm going to use are these cases. Again, today they will look different. They will have different sizes, different lengths. You're right. That is still local. But I'm looking for the local ones that are unique, at least in today's database. And you would be right saying that maybe in tomorrow's database they will no longer be. That is true, and that is a possible problem. But let's ignore that problem for the time being. It is an important problem. That is a good answer. But again, my algorithm proceeds the following way. I find fragments or consecutive runs that are unique in today's database in one of those three states. And then I have my set. I simply take my protein, my new sequence that comes in, and I look up in my database, is one of those match criterion, right? If match criterion, I call that helix. Would that work? Again, let me repeat to you. It's absolutely clear that you do not know the answer. You are here because you want to learn about it. So you cannot know the answer to my question. The game is about guessing or coming up with right or wrong answer. I don't care. What is important is that you learn to argue.
I would guess it would work for all helixes that I know. So
yes, that's one problem, yes. So it would work for everything that I know. It may not work for everything I don't know. That is true. That's the first statement, and that is very, very clear. But it may work for some things I don't know, right? But here's another problem with it. It will sort of chop my protein. I will match, and then I can say something, and then I will have many states where I don't match. So I will not be able to say anything at all. So essentially I translate the three-state problem into a four-state problem, where the fourth state is I don't know. Right? That is one thing so I essentially have two problems. One is that I don't know how that is going to work tomorrow. And the second problem, this is clearly not going to work everywhere. Because tomorrow means that some structures are not in today's databases. So it may work for those in between, but somewhere it clearly doesn't work. But actually the first method was much simpler than that. The first prediction method that was published. And the first prediction method, setting the stage, Linus Pauling in the 50s, that went to a Nobel Prize, Chengerji had a science paper in 57, in which he proposed the first method that predicts secondary structure. That method simply says whenever you see a proline breaks a helix. If proline breaks a helix, then proline is in a helix. So when you see a proline, you know there's a helix. That was as much as this method essentially did. The remarkable part of that is that the first structures were done afterwards. So we had the first 1D prediction methods prior to ever having observed secondary structure. So in 57 somebody predicts secondary structure, in 59 or 60 somebody observes secondary structure. This is remarkable. So it predates the actual structures here. Now, again, what I said, the proline is shaped in a way in which it bends the backbone, it bends the chain, and this bend leads to breaking a helix. Now, bless you. How can we go from the simple idea of proline breaks the helix to something that sort of involves all 20 amino acids? So I have two simple methods now. One is sort of finding k-mers that I looked up in a database and will not always work. Another one looks only at proline, and for 19 of the residues it also doesn't work. So we have two very choppy methods. Can we fill in at least the proline type of idea for a complete method that has a prediction for every residue? Yes, we can. The question is how. So you have 20 amino acids. I give you data, and you want to predict the secondary structure state, one of three states, HEL, or HEL, I'm just going to use that, for every one of the 20 amino acids. How could you do that?
Every amino acid, the R group is unique for every amino acid. So we can categorize them into which falls into helix or
How? So let me repeat.
based on the R.
The side chain, yes. So based on the side chain, I can possibly classify my proteins, and I can classify in a way that would allow me to do this. That is true what you say, but it's not explicit enough.
Classify into structures, not in the sense classify based on side chains into helix,
Yes.
trans, and others.
But how am I going so classify side chains into alpha helix, beta strand, other? But how am I going to do that? Look at it? You're going to look at the side chain, so I'm going to give you the side chains again, and you tell me which one is going to be in a helix?
No.
No.
We have the structure already, right?
Yes.
We know which amino acid is going.
Yes.
So based on if it is a helix, we know which amino acids or the side chains are there.
Yes.
Right? So we can give a probability to each of the side chain that if it is there, it will be a helix or a helix.
That is right. Let somebody else take over. So you're going in the right direction, but I want somebody else to be more explicit. So so this is the typical problem that I have as a teacher. When is it that you blah, and when is it that you really know? I believe at this point I know that you know, but I believe it's still too early for the others to have understood what you said. So my question now to the others is, how can you do this? So again, the question is, how can we go from profile breaks the helix as a rule to something that is a more generic odds for all 20 residues? How can we do that? Anybody? Come up. You have 20 amino acids. You have a database of, for every single amino acid, you know the secondary structure in which it's observed. Sometimes the same residue is observed in two different secondary structures. But what can you compute? Hello, I give you a distribution of 20 letters, and I give you a distribution of 3 states. What is it that you do first? You need whenever anybody gives you data, begin to do something with it. Well, first you look at it, but then, first you parse it, then you look at it, but then begin to look at some aspects of the data. Whatever those are. Immediate question that you would ask is, somebody gives me 28 letters, did they actually really give me all 20? And do they come at the same frequency? Or somebody gives me 3 secondary structure states, is it really 3? Do they come at the same frequency? And these questions is teaching you something about the data, whatever data you have. But it's also teaching you whether your parser is right. Very often this is how you find programming mistakes. That a profiler doesn't find. Okay, profiler is outdated. Anyway, so I essentially have everything here now. How can you put 1 and 1 together to get a 3 out of it? I have to learn to communicate to you guys. Somehow we're not doing well. That's your sequence. That's your secondary structure, where here I have dots, but usually where there is an empty space, that means the other state, the loop state. again, here's a table with the sequence that I copy from there. Here's the secondary structure, L and E. In this particular case, we have only 2 states. And you simply count for every single letter. How often does it occur at one of these 2 or one of these 3 states? In this particular case, everybody on the 3rd state gets a 0. And you do that for all the data set. And that gives you log odds. Very, very simple. Gives you a simple probability. That's a very simplistic method. It may not work, but it's the best you can do in 10 minutes. Or whatever time it takes you to parse the data. From that moment on, you immediately have it. And it is a simple prediction method. And the next question then would be, how well does it work? And in fact, that is essentially what the Chow-Fussman GOR methods were. They did exactly that. And they are called Chow-Fussman, the authors here of this paper in 74. GOR is also author papers, is the initials for the last names of Garnier, Osgothorpe, and Robson. And that got the name GOR. 78 is the latest implementation of that. And the statistics really came from Barry Robson here in the 70s. And again, the simple thing it does, it compiles a probability for particular residues to be in one of the 3 secondary structure states. Yes? In the top row is what?
Is the 20 letters you gave us?
Yes.
In the table?
Yes, it's not 20, is it? So several are missing, yes, but these are the 20 amino acids, yes.
And these 2 are the states?
These are the states, yes.
This E and L?
Yes.
So does it mean like one amino acid corresponds to either helix or loop or a strand?
In this particular case, I'm sorry, thank you for interrupting me. I should have made that more clear. So again, sequence is 1D, secondary structure is 1D. Therefore, I can essentially present one over the other. So far, I've always shown you secondary structure as a color in a 3D structure. I've shown you the helices as cylinders and the beta strands as arrows. Now, ultimately, this is 1D. So ultimately, in this particular case here, I do not have the information how things hold together. All I know is the sequence of secondary structure symbols. And that's a 1D thing. So for residue 1, 2, 3, 10, and so forth, 22, 3, 4, for 24 residues here, I show the sequence and I show the observed secondary structure corresponding. So the first 6 residues here are in state other. And then comes a strand.
How do you find it?
This is the lookup from a PDB structure. So this is a lookup from an experimental structure, which I have a 3-dimensional, in this particular case here, I have the 3, essentially this is the question that you're asking here, which is was going to be the next slide before you made me go backwards in this, but thanks for asking. Once in my life it fits. Usually this is not the case. So where do we get the secondary structure from? And essentially we take a bunch of proteins for which we have experimental structures. We apply a method that takes 3D structures and converts it to 1D structure, secondary structure, such as DSSP, that given as an input 3D structure, writes secondary structure out in 7 states here, and we boil down these into a 3-state helix, strand, other, alphabet. So the resource with 3D structures of proteins and RNA and DNA is the PDB, the Protein Data Bank. PDB has, that's the URL, it has aspects such as the molecule of the month, and it's sometimes really cool to read it. This month, this is from David Gotzel, this month is about titin. So this is the way titin look. It's a huge, this is the longest proteins that we have in human, and in fact the longest in the universe that is known, 4,000 residues. Remember, some proteins are 100 residues, some proteins in fact are 60 residues. This has 34,000, and it's just a giant. Now, oops. In terms of cumulative numbers, you see that about 1% or much less than 1% here, half of a percent of all the eukaryotic proteins, and less than that for all the other proteins, have over 2,000 residues. This has 34,000. So this is the odd one out of the odd one. Exponential, this is a logarithmic plot. So PDB explodes. Now, again, we take a unique subset from the PDB, convert 3D to 1D, and get secondary structure using DSSP. And then, essentially, we have the label, the label for the states of the first method. In fact, the first method I said was Gore or Chow-Fussman here. That is about at this time here, when we had about 80 proteins. So for 80 proteins, you have this assignment, and then you can compile the probability. That brings us to the next question. How do we assess performance? How well do we do? How can we assess that? Give me numbers. What could you measure? And again, to make it simpler, here's the problem. In this particular case, this is not amino acid anymore. What I have here is an observed string and a predicted string, for instance. How can I measure the agreement, the performance of my prediction method? Come up with a method. Give me a measure. Can you speak up a little bit?
What is the difference between observed and predicted?
So observed is essentially what I have in the database. Predicted is my prediction method. So we have now, assume this would have been Chow-Fussman or Gore, the first Gore method. So simply we take the residues, look at the probability from the observed strings. Once we have done all of that, we have a probability assignment. So we're not looking up, like the method that I talked about, the K-mer method, we're not looking up in the database, is that a helix? What we are doing is, we have a certain probability for each of the 20 amino acids to form a helix. And then we have an observation of that particular residue, and according to the probability, we have a prediction. Okay, we simply take the state that has the highest probability. That's the simplest way of doing it. There are other ways. If you could smooth out a window, you can think about other ways of improving over that. But the simplest one is really top wins. And in the top win example, you have a prediction. And that is the second one here. So the first string is the observation, and the second one is the prediction. And even for the data set that you learned from, since this is such a simple model, such a crude model with a probability for every single residue, you will make mistakes on the training set. Because you don't have that many parameters, 20 parameters, right? Essentially. So, question again, we have these two strings. How can we measure their similarities? One way of looking at it. Another way of looking at it, how can I assess how well my method works? There are different ways I could do. Just throw some at me. Try the first. Yeah?
Number of correct matches.
Yes.
Minus the incorrect matches.
Oh, that's an interesting idea. No, but wait a minute. So, in this particular problem, the sum is a zero-sum game again. So the number of correct matches, I don't need the number of incorrect anymore. The number of correct completely defines my problem, right?
Yeah, but
It's a percentage. The total number and the number of correct, and then you can compile the mismatch. Just in the same sense of the not well-defined loop state. Yeah, no, now you have to be more explicit. So the first statement, I understood you to say, we just look at the percentage of agreeing letters. Yeah? But now you're going in a different direction.
That means when I said minus, I meant I want to put some penalty on
How?
correction also.
Yes, but that is exactly implicitly a penalty. The percentage of agreement is a penalty for disagreement. Isn't it? You could think about something else, but you have to be more explicit if that is what you're thinking about. Any other method? Look at what you see on the screen. What you see on the screen is a method, and I will get back to that later, is somehow a prediction that is chopped up in some sense, that has short fragments. So again, when I showed you these images, you saw these long cylinders. You didn't see a little bit of a cylinder, a little bit of an arrow, a little bit of a cylinder, a little bit of an arrow, as you see in this prediction method here. And you saw from these images that, so where are the cylinders is somehow what you're after. You're not necessarily after every single residue, whether that is a cylinder residue, but you want the whole thing somehow. And you could try to measure that. So you could try to define segments. You could try to introduce a method that simply says, I can predict that helix. Not, I can, or in this particular case they are strands. Not, I can predict of these 6 strand residues, I can predict 2. But do I get the strand essentially right or not? And there are different ways in which you can do that. You could say, since the begin and the end is predicted, that looks like an overlap. Or you could say, no, that's not enough, you need a minimal overlap. In this particular case, most possibly you would say that in none of these cases you have a minimal overlap. Or, again, depends on the way you define your measure. So what you said, you explicitly penalize the mismatches. This you can carve into an entropy. As a computer scientist, you have certainly seen Shannon's entropy. And you can simply use this to compile a Shannon entropy type of measure, a mutual information content, or whatever you call these things. Essentially, it's an energy function, it's a simple information index that, in fact, in some sense, penalizes. That begins, so the difference between a percentage that implicitly also counts mismatches and Shannon entropy is that the Shannon entropy explicitly requires, or is built into the formalism, that you have 2 states and you want to match 2 states. Here in this particular case could be 3 states. It's a little bit more complicated to do this. But you can still define an information content for that problem. Okay. That method originally was published for the first time in this book here, and was called Q3. It's the quotient 3 for 3 states. This is not quite what the authors meant, but this is sort of an interpretation of the word Q3 that makes sense, where you simply count the number of correctly predicted residues and state helix strand other, 3 because of 3 states, and then you divide by the number of residues. Okay. So what was published for these first prediction methods that simply look at the individual residue probability was that they would reach 63%. Okay, the first question now, whenever you get a number for a method that you have, is that high or low? And if you have other papers published, then you can easily answer that question. But if you invent something new, truly novel, then you don't know. You cannot compare yourself to anything because you're new. So, this is new to you. Is that a big number or not? Is it a good number or not good number? Yes is a good answer. Yes is always good. No is, for a scientist, is always good too. But why? The real question is why.
Because in 63, you don't know the structure of the protein. You are predicting it after seeing the sequence. So 63% is for me it's too much.
63% is too much, or?
It's too much.
Too much. So you believe they're lying? And I'm a little bit confused. What do you mean by too much? Do you mean that I'm lying? Again, so I have several parts of my talks where I put in lies. I typically try to scratch them out such that you remember that they were lies. But yes, this has happened before. It will happen again. So it could be. Is that what you're saying? No, it's not what you're saying.
My point is, like, if your result is 63%, then your experiment is very good.
Oh. So you believe this is a high number. Okay, 63 is a high number. Anyway, it's a round number. 6 is good, 3 is good, all is good, and we are happy. But I want an argument. Why is 63 high? Let's begin there. No, can I have somebody who has, yeah?
Could you compare it with the random prediction?
Yes, that's the important statement that I wanted. We need to put this in background. We need to see this behind the light of random. And random simply is throwing a coin. You have, and what would the coin give? If we had an equal distribution of HEL, what's the random giving? 33%. So, behind random, 63 is more than double. So that is a very, very important question to ask for whatever you do. Put the random background. You can always do that. And you always have to do that. Whenever you ask yourself, did I do something right? The first question is, what is random? Okay, so behind random, this looks good. Now, let me go first into a different detour. So, the way they had analyzed that this is a method that is done in the 70s, remember the secondary structure assignment from DSSP was in the 80s. So the prediction method that I talked about was before there was an automated method to assign secondary structure from 3D structure. How do you believe people did it before? So we had a time when we have 3D structures, but no method, no automated algorithm to take these 3D coordinates and spit out a classification to secondary structure. How do people, and people have developed secondary structure prediction methods, so how do they get at the labels? How do they get at the data? Which I understand was your question before, and I sort of tricked you into saying you use DSSP. But those days, in fact, you could not do that. So how do you believe they did it? So they just stared at it. So crystallographers, so people who did the first structures, they simply look at the structure and say, this is a helix. My expertise says this is a helix. And that is the classification that has been used to come up with the 63. When the same method was filtered through a larger data set and automated assignment by DSSP, this was in fact the result. So the method actually, so that was a lie in some sense. 63 turned into 50 to 55, somewhere in that ballpark, and it's closer to 50. Which is still, random is 33, or since the states are not equally distributed, random actually is 35. So still clearly better than random. Now, we could improve over that. This, in fact, is the first generation method. There is a second generation method where there's a first generation. What do you believe you can do? Well, how can you improve? Yeah?
You can measure the frequency of the surface protein and compare the frequencies between the predicted and the observed.
No, I didn't get that. Let me repeat what I believe you said. You can look at the frequency, the difference between the frequency of observed and predicted secondary structure. So you can essentially correct your method if you overpredict helices. That's an interesting idea. Is that what you want to say? So you could actually, so what you're suggesting is we do what we did so far, but then we say at the end of the day that we overpredict helix, so we're going to correct for that overprediction. Is that what you're saying? That's a great idea. And in fact, that is an idea that I don't know why I've never seen anybody have in the olden days. It's a very good idea. I do actually not know why they didn't do that. There's absolutely no reason why they didn't, because so I have clear data that indicates that this would have worked a little bit better, clearly. So they clearly could have improved their method. I know that. But they actually never did it. I never thought about it either. Great idea. But I meant something bigger. So bigger in terms of algorithm or methodology, or in terms of the idea of what I use as information. But so what I like in this idea, some of you I do not know whether there's anybody in the room who sort of grew up in Germany. In Germany there's a character, he's called Münchhausen. And it's a Baron Münchhausen. He has all kinds of strange stories that he's telling people. One of the stories is that he's riding into a sump with his horse, and rider and horse go into the sump and essentially go under. And then he pulls himself and the horse together out of the sump on his hair. He obviously has hair like I, and that's why I have long hair, right? So that one day when I end up in the sump, I can pull myself out. But somehow this is a similar idea. You pull yourself out of the sump because you don't use new data. You don't bring in anything new. You just look at what do I do wrong, and you do an aftermath correction. And I like this idea also because this is something that you can actually always do. And in this particular case, it would have helped. Okay, great, thank you. But I meant something more different than that. A different idea. So far, what we have is really, we look at every single residue and ask what's the probability of being in a helix. And what you say would correct for that overall, so I would change the threshold. Essentially, you introduce a 21st parameter. But essentially, still is the same sort of 20 parameters. Is there a way to sort of blow up the complexity a little bit more and use some bit of information that you may know matters, or that by now we all know one residue is not enough. We also already know that pentapeptides are not enough. But clearly pentapeptides are already better than so far what we have here in the first generation single peptides. Or can we improve over single peptides, or single residues, single amino acids? I have a probability for a single amino acid to be in one of the 3 secondary structure states. What could I do that is already better than 1? Yes, 2, 3. How would you do that? Anybody else? So a number that is higher than 1 is 2, or 3, or 4, or 5. Let's look at the pentapeptides. How would I do this with a pentapeptide? How could I, in fact, use a consecutive stretch for a prediction method? There are 2 principal differently ways in which I can do that. One we already talked about. Could you repeat that? I understand that I'm hiding the fact that it's not that obvious. I understand that. Think about words. Okay, misleading. About things, the meaning of what I mean by word is the meaning of a word is not necessarily captured in a single letter. Very often I can write things down and even make a typo, and you may not even recognize because you just don't see that. You recognize the word. Your brain is optimized to quickly reading and to quickly getting whole stretches. And in some sense that's what I mean here. Segments. Yes?
Thinking about words, maybe trying to have some patterns of like syllables.
Yes. So, sure, somebody who speaks Spanish must say that. So patterns who are syllables. So some continuous stretches of words that in fact make up the Spanish language. That's why you guys are so fast in speaking, because you're very good at that, matching syllables. So, yes, and that in fact is exactly the idea here that Kapsch and Zander, when they did their secondary structure assignment method that made them famous, it's quoted over 10,000 times this paper, that is not what they wanted to do. What they actually wanted to do is syllables. They wanted to identify words that map. So remember, these are the guys who also saw that there are pentapeptides. So essentially they had exactly the idea of coming up with a dictionary of syllables that they can match to secondary structure states in a clever way. But for that they needed a data set that was much larger than the data set they had. And for that they needed an automated assignment method. And they ended up, instead of publishing, so they wanted to predict secondary structure. There's a preprint, but this was never published because they ended up doing the assignment method and then it went completely different directions. But that was their idea. It took many, many years before the first one picked up the same idea. Here is the second way of doing it. And that second way comes from an algorithm that is in the literature as GOR3. It's the same group of people, published a little bit over a decade later. It's the GOR method. It is called GOR because it's the same people, but essentially it's also the same idea. Now we again look at the probability, odds, and that is not the syllable idea. Now we get a pure probability, but now we have two things. We have a probability of the central residue here, or we have the central residue, and we ask what's the probability of this residue being in the helix given that 3 to the right and 3 to the left, we see these other 6 amino acids. So it's a conditional probability, conditioned upon my environment. And you see already in the first attempt they were bigger than pentapeptides, because at this point they already know that pentapeptides are not right. This is after the pentapeptide is in the Helix and Strand publication. So we are already going into 7 mers, into something that at this point in time people had not observed in the same state twice. Okay, so how well does it do? It reaches levels of sort of 55 to 60% accuracy. The problems with those methods, in fact, it's not 100%. And the argument that was around at the time was, well, 100% we cannot reach because we use only local structures, and only 65% of the secondary structure is defined by local stretches. So does anybody have an idea how you can come up to the number 65? So I know how to get it, but I don't know how to get it to you. I'm not good at jumping. So how would you get it? Why do you believe this number here is 65? So again, the number here, 65, is that people claim that only 65% of secondary structure is defined by local stretches of, for instance, 11 consecutive residues. How do you believe people come up with a number like that? Yes? Somebody?
It's maybe because there are 3 structures, helix, strands and loop. So if we take equal distribution, 2 by 3 makes 67.
Guys, I have a problem now. You do exactly what I say you should do. But now we get into voodoo. How can we distinguish between science and voodoo? What you did is voodoo. So you explained the 33. And the factor 2 came as voodoo, factor 2, give or take, who cares, right? So I have the 66. But that is a big jump. If I explain 33, to then make 33 suddenly look like 65 is a stretch. And that is the type of stretch you have to be careful with. This is the type of thing that gets us, again, not you. That is what we as scientists very often do, because we learn it's true. Often the factor of 2 is not important. So put it in or take it away. So we have our experience, this is the kind of thing that we learn to do. But in this particular case, it's really a mistake. Because there is no logic for that factor of 2. So again, what could be the reason for the 65? And let me repeat, saying things that are wrong. The only way in which we proceed as scientists is by making mistakes. By always doing it right, we cannot advance. That is, as long as we're always right and we always do the right thing, we stay exactly where we are today. We will never advance. So you need to make mistakes, so just come up with guesses. That's also voodoo. I'm calling that voodoo because you essentially do something like that. So you have a number that you observe, and you have a number that you know. And now you combine the number that you observe and the number you know in a way in which arithmetically you would end up at the same number. So again, for people who haven't heard that here on the left extreme, he rightfully says that essentially, so what we random is 33 or 35, given the non-equal distribution. 100 minus 35, here we are, bingo. Right? Sounds totally convincing. And since it's so much on the dot, you in fact may be inclined to not really worry or wonder why. So why should you subtract random from 100? And what we have is a random, which has nothing to do, the random value has essentially nothing to do with local versus not local. So logically, these numbers are in fact not related. Again, this is exactly the kind of thing I wanted you to do. Thank you for doing it. In doing science, you have to, this is the process that you have to become more aware of. Put things out in front of you, typically on a piece of paper or on a computer. Sometimes think about it overnight. Come back to it after a week, come back to it after 3 months, and query, ask yourself. This is the scientific process. So no, because that answer is not right, because in fact these are unrelated logical numbers. Random subtracted from 100 does not tell me anything about what is local. So the answer to the question here essentially is that people developed all kinds of prediction methods, and none of these prediction methods hit that threshold. The conclusion then was, well, obviously there's only, to some extent, the local secondary structure defines only 65% because we cannot get longer. And that, in fact, again, is a way in which scientists and science proceeds very often. You try to hit down a barrier, and once you cannot, you essentially say that is a real barrier. And then you begin to explain why that is a real barrier, and why it's not you who is a loser not to be able to get over it. Okay. And again, so the logic here is very convincing because helix formation is local. That works. But beta strand formation is not local. Actually, what would that mean in terms of predicting beta strands and beta and alpha helices? Do you believe they are predicted equally well? So one is stabilized by local interactions and beta strands are stabilized by non-local interactions. Do you believe prediction accuracy is similar for them? Who believes it's similar for beta strands? Don't look around. The belief is in you. And crowdsourcing is very, very interesting information, but it's not always right. We had at least 2 examples where the only one who was the exception was right. Again, who believes that beta strand and alpha helices are predicted equally? Who believes they are not predicted equally? Yes. And then in this particular case, crowd was right. So beta strand, in fact, that is another way of looking at the question of how can we assess prediction accuracy, prediction performance. One is the overall percentage. The other one is the percentage differentially for the states. So is helix predicted better than strand? It's not what you see in the overall percentage. And when you look at the beta strand propensity, then you see that one is in fact 40%, which is in some sense close to random. And the argument was exactly that. It's not as local. And that all made sense. The next problem of prediction methods was that segments tended to be predicted way too short. And that is exactly the example that I showed you before. A typical prediction method of the first 2 generations would have this as a predicted string, that being the observed string. And you see, we had that before. In this particular case, you don't quite know, do you somehow see that there is a secondary structure segment and you get it right? Or do you get it entirely wrong because that is actually helix? In this particular case, is this what you predict here, one long beta strand? Or is it 3 different strands? Is this a helix and a strand, or is it none of the above? And the same is here. So if your fragments are too short, you don't quite know, as somebody who uses them in the aftermath, what to do with it. what to make out of it. What else could we do to predict secondary structure, to improve over what I showed you so far? So far, this is all conditional probabilities, but it's relatively simple, right? Any other idea? So far, I talked about methods that were published long before most of you were born. Okay, I was wrong about this sort of thing before. Was anybody born in the end of the 70s, beginning of the 80s? Nobody was, right? Okay, I was alone then in this room. So this is all before you were born. What is more contemporary? What is more modern? What could you do? Something, in fact, that came in the 90s, or became strong in the 90s.
To predict?
To predict, yes.
Like neural network?
Yes. So in machine learning devices, exactly. With machine, so far, what we have is a conditional probability. A conditional probability is a very simple methodology. You can put that into a Bayesian network and already it gets better. But there are tons of other things. And what neural networks, for instance, do is they look at bunch stretches, at similarities. They're very good at looking at patterns or other machine learning devices. There are machine learning packages. Weka is an example for that. And if you have not seen that and ever encounter machine learning, Weka is just a nice toy to begin machine learning, playing around with machine learning with. A neural network historically was the simple thing, because this is the kind of thing that you can program in a few minutes. Well, not a few minutes, it takes 20 minutes and you can program your own neural network and then play with it. And that is fun. So let's begin with the concept of the neural network, and after that I will close for today. So neural network is taken here just as one of the early methods for machine learning. What a neural network does, or one of these particular types of networks, are these layered networks that are as follows. You have in blue an input, and in green an output. You have a connection between these two in this way. The computation is done by you multiply the input here with the connection, with this value. You do the same here. This unit actually sums over this product. So input times J12, input times J11. You add these two, that comes into the left side of the green unit. Out comes a functional filter of that. Now, what the first part here does is you have two vectors. You essentially multiply two vectors, and what you do essentially you project one vector onto another. Geometrically, that's what this machine here does. The input to here is just a projection of a vector. What the trigger function, such as the sigmoid here, or the hyperbolical tangent shown here, but it's the same. Just any sigmoid function. What this one does, in fact, it answers, is that longer or shorter than something? Okay, so it has a switch. For very high positive, it goes to 1. For very high negative, it goes to 0, and something in between. Okay? And that makes the neural network become a non-linear device. The way you train it is very simple. You train neural networks with examples that you have. So so-called supervised learning, meaning you have examples for which you do know the answer. In this particular case here, for secondary structure, you have sequences for which you know the secondary structure state. So say you want to classify the open circle and the dark circle here. In this particular case here, what you have is a vector of 1, 0. Now, the mistake that you make is simply what, doing your computation here, comes out minus what you expect, and then you square that. Clear, right? How do you train that device? The free parameters, everybody sees what the free parameters are? What can you change in the game? Okay, let's go back to, that's my neural network. And what does it mean to train it? To make it fit to data? Can anybody? Sorry? These or these? The problem is, you may absolutely say the right thing, I just don't hear it, I'm sorry.
There are some numbers that you multiply to the output.
Just which, do you see a symbol? Or do you see anything?
One and two, or
This?
no.
No, but that's the input.
Equation, right.
In one, that's the input. You cannot change that.
What about J11 and J12?
That's exactly the answer. So yeah, by exclusion principle it had to be the other one then. Yes, so the connections here, that's where the free parameters sit. So you can make the connections fit to the data such that you can learn the entire data set. And the way you essentially do that is, once you have this error function, you have some connection space related to some error. So you begin to initialize with some random values for these connections. You cannot have them 0. They need a value. There's a reason for that. You pick a random distribution, and from that random distribution you make a certain error, and then you simply go downhill in that error function. That means you essentially change the connection every single time that you present a new example. You change the connection in a way such that the error gets lower. Okay? And that is sort of one by one how you change it, and I'm typically too lazy to go through this example. But this example is constructed such that you, for this particular XOR problem where you have a 1, 0 and 0, 1 that you want to classify, and they come in, how step by step you change the connections. And a simple example. What you can do with that is you can classify these in this XOR. You can classify these two types, the open circles and the dark circles. And these are lines that the neural network, because essentially it's a vector, you multiply two vectors, you project two vectors, essentially you introduce a line. And left or right is the hyperbolical tangents. That is left and right of the line, essentially, the meaning of left or right of the line, or above or below in this particular case. The different lines here are all solutions that in this particular case do it. Okay? What about the other problem? Can you show me the way in which you can separate out the points here with a line? At least one person says, no, I cannot. I refuse to do that. They're not thinking out of the box. Sure, you can show a line. It's a perfect line. I never said a straight line. This is the kind of thing that we do all the time. In science, try to sort of, this is again, that's the most complicated part. Going back to what you assumed so far and what of that doesn't apply. Anyway, so you could have this line, but that line does not fit in the language of the neural network. In the language of the neural network, the only way you can do it is straight lines, and the only way you can do it with straight lines is you need two. And then you say whatever sandwiching is inside between these two lines is an open, and everything outside of these two lines is dark. That you can do. But now in the language of the neural network, how do you introduce a second line? So far what I showed you, there's a line, full stop, not a second one. And the way you do that is essentially you introduce another layer of neurons. This other layer in principle allows you to, in this particular case here, give a second line, and then the story gets more complicated from then on. At this point, what I show you, all the analogies are still strict, but the moment you blow up this hidden layer, so the add hidden units, that moment you go into hyperspace, and then 2D and 1D no longer matters. So essentially what you then do is you have a hyperspace in which you distinguish with hyperplanes. But other than that, it's still the same analogy. Still the same 2D analogy. Everything simple, it's just you can't really compute the dimensionality easily anymore. You cannot change the dimension of this one because that's the input, and you cannot change the output. Now, the way you simply do this is the same as before. You take what comes out here, multiply it with that connection, that's the input here, sum this product here, then you apply the trigger function, do the same thing here, and do the same thing again. You multiply the output here with that value, do with that value, add these two products, is the input here, trigger function, output. Same as before. Totally the same concept, it's just a repetition of the same, and that's the formula that looks a little bit complicated, but it's totally trivial. And just like before, you simply then go, you project vectors, and you go, in order to train that, you go downhill in some error landscape, and reduce the error. What you have to avoid, what I show here, is training time. That is performance, accuracy, and these are two data sets. Green is the training set, and blue is the data set that I actually want to see whether I generalize. Whether you said that before, if I do my segment method, maybe it works for proteins I have in my database today, but not for those that will come tomorrow. The blue one here is supposed to estimate the performance for proteins from tomorrow. And overtraining essentially means that you perform at some point, the training helps you only for the green data set, but not for tomorrow. So that is what you call overtraining in machine learning, that's what you call overfitting in statistics, and that's what in all walks of life you see as something that you have to avoid. With neural networks, this is particularly complicated to avoid because you can blow up the number of hidden units. You can blow up the system such that you immediately overtrain. The huger you blow it, the more free parameters you bring, so the less data points define your free parameters, the more likely you will overtrain. The more complex your problem, the more likely you will overtrain. Okay. I believe I will stop here. Before I stop, are there any questions? Thank you.